Hi everyone, welcome to our topic 4, meiosis review. Okay, so meiosis is the process by which gametes are formed. Okay, so now we're asking, okay, well, what are gametes? Gametes are sex cells or cells specialized in passing on genetic information to offspring. Okay, so this would be their function. Females produce female gametes, which would be ova, egg cells, or ovules in plants, while males produce male gametes, such as sperm or pollen. There are some species which are a combination of male and female, so a lot of plants have flowers with both male and female gametes. Gametes, how are they formed? In a process called meiosis, okay, which we just talked about. So in case you were curious, in a flower, the flower anatomy, we have a stamen, and this is where the pollen is located. And where does fertilization happen in flowers? So this is the pistil. The pollen would enter through the pistil, and down here to the ovule, this is where fertilization would happen in flowers. So meiosis, if we look at the process of meiosis, what part of cell theory connects to meiosis? So is an organism able to just make gametes? Well, no. They have to use pre-existing cells to make gametes. So we have to start with a pre-existing cell to produce the gametes. Okay, so that is what this slide is about. What part of cell theory does meiosis relate to? All cells arise from pre-existing cells, number 2. Okay, so we have some new vocabulary here. We have haploid and diploid. So if a cell is haploid, that means it has one copy of each chromosome. If a cell is diploid, that means that it has 2 copies of each chromosome. For example, this would be a karyotype of a diploid cell. So for each chromosome number or chromosome type, we have 2 homologous chromosomes. A haploid cell would have only 1 chromosome of each type. So homologous chromosomes are 2 chromosomes with the same genes. So they code for the same proteins or the same genes, but they might have different alleles. Okay? So homologous chromosomes, same genes, perhaps different alleles. Okay? Most cells in the body are diploid. So we have somatic cells are diploid. Okay? Our haploid cells would be our gametes. Now, why do gametes need to be haploid? Gametes need to be haploid so that after fertilization, the resulting offspring is diploid, just like its parent was and its grandparents were. So if gametes were diploid, then our resulting offspring would be like quadruploid. And then the one after that would have 8 of each chromosome. And these would be huge abnormalities, right? So 2 haploid gametes after fertilization would produce a diploid offspring. And so my other question is, what exactly is fertilization? Fertilization is the joining of 2 gametes, the genetic information of 2 gametes. Okay, so pollen and ovule joining their genetic information to produce a seed. Okay. So now if we take a look at the process of meiosis, we're starting here with interphase with a diploid cell. If you look carefully at this cell, we have 2 pairs of homologous chromosomes. We have a short pair and we have a long pair. We can distinguish between, our homologous pairs should be the same length as each other. So if you look at a karyotype, if you notice, the longest chromosome is number 1 and the shortest chromosome is number 2. So when we design a karyotype, it always goes longest to shortest. And homologous chromosomes should be the same length because they have the same number of genes on them. Okay, so if we are trying to identify the homologous chromosomes here, we're going to identify them based on length. So the long ones will be homologous and the short ones will be homologous. Okay, so this cell at the beginning here is diploid. okay? And our cell stays diploid. So now we go into prophase 1, prometaphase, metaphase 1, and the chromosomes are now aligned down the center of the cell. Homologous chromosomes are paired. Okay, so my long chromosome pairs with my long chromosome, my short chromosome pairs with my short chromosome. So maybe you might be confused, Miss Lisa, why are they like X's now? Why are they like X's? That's because right here, DNA replication happens. Okay, so DNA replication happens. So when DNA replication happens, now our chromosomes have a replicated version that they are attached to. So here is my original chromosome, and now it replicated, and I have two identical copies of my genetic information attached to each other. Sorry. So my question is here, is this is a karyotype of prophase 1. Is prophase 1 haploid or diploid? Prophase 1 is still diploid because I have one. Sorry about that, my laptop died. So this is a diploid cell because we have 2 chromosomes. I realize we have 4 copies here, but we only have 2 chromosomes. So this is considered 1 chromosome because it's connected at the centromere, and this is a second chromosome because it's connected at the centromere. Okay. All right, so here we go. We have diploid, diploid, diploid. In metaphase 1, our homologous chromosomes are paired, and actually what's occurring here is crossing over. So this is where crossing over occurs. And you can see here, now I have a little bit of blue on this chromosome, a little bit of red, a little bit of blue on this one, a little bit of red on this one. So by anaphase 1, crossing over has finished, and my homologous chromosomes assort independently of each other into the 2 resulting cells. So they are independently assorting themselves into the 2 cells and then going into prophase 2. In prophase 2, this cell here is haploid because now I have 1 long chromosome and 1 short chromosome. So 1 long and 1 short. As I continue down, we still are haploid. My chromosomes are lining down the metaphase plate. Okay, still haploid. Then in anaphase, my sister chromatids assort independently into the 2 resulting cells. So it could go either. So if this one here, it's hard to see, but this one has a little bit of red on it. This one doesn't. So this one with a little bit of red, it could have gone to the left or it could have gone to the right. This is independent assortment. It's random. It can go either direction, okay, but they assort independently of each other. And in the end, we end up with 4 genetically different gametes. Sometimes, though, there are mistakes during meiosis. One of those mistakes that can happen is non-disjunction. So if non-disjunction occurs in meiosis 1, that means that my homologous chromosomes fail to separate. So I have 1 pair of homologous chromosomes, or both pairs of homologous chromosomes end up in the same cell. And what happens is 50% of my gametes have an extra chromosome, and 50% of my gametes are missing a chromosome. Okay. What would happen is these gametes would result in the offspring having a monosomy if they were fertilized, and these ones would result in a trisomy if they were fertilized. Over here is the second case scenario where the homologous chromosomes split perfectly in meiosis 1, but then in meiosis 2, the sister chromatids fail to separate in one of the cells. When this happens, then we end up with 1 gamete that has an extra chromosome, one missing a chromosome, and then 2 normal gametes. And some disorders that we learned about that result from non-disjunction would be Down syndrome, where we have a trisomy. So one of the gametes contained an extra chromosome number 13. Okay. We have Turner syndrome. One of the gametes was missing a sex chromosome. So it could have been missing an X or a Y. We don't really know, but it was missing a sex chromosome. And in this case, with Klinefelter syndrome, one of the gametes had an extra X chromosome or an extra sex chromosome in it. Okay. All right, so let's continue on. Crossing over. So during metaphase 1, okay, my homologous chromosomes come together like this, and they cross over some of their genetic information. So some of this maternal chromosome went attached to the paternal, and some of the paternal attached to the maternal. So they switched, okay? And normally this should happen perfectly. They should break at the same spot and switch at the same spot, and there should be no issues in this process. However, sometimes they do not do that correctly, and that would result in some of these chromosomal mutations we see on the next page. But let's talk about linked genes for a second. So genes that are close together on the chromosome, it's very unlikely that the crossing over will occur between them because it's a very small area. So the smaller the area between two genes, the less likely the crossing over will happen between them. The greater the area, the more likely crossing over will happen somewhere between them. Okay? So in this situation, we have 3 genes. Where's the crossing over most likely to happen? It's most likely to happen between, it's very likely to happen between Q and R. It is decently likely to happen between R and P. And it's unlikely to happen between P and Q because they're the closest. So I'll say this again. Crossing over is most likely to happen between blue and yellow because they're the furthest apart. Somewhat likely to happen between, blue and green. And least likely to happen between green and yellow. So genes that are very close together are called linked genes because they are likely, the alleles are inherited together. So for example, if my maternal chromosome says blonde hair, blue eyes, and my paternal chromosome says brown hair, brown eyes, when I'm making my gametes, I'm probably either going to put the brown hair, brown eyes, or the blue-eyed blonde hair, okay? I'm probably not going to put blonde hair with brown eyes because crossing over and that shuffling of alleles is unlikely to happen between genes that are close together, like eye color and hair color, okay? So these are some of the chromosomal mutations. And we said that this one is deletion because part is missing. This is inversion because we have it flipped, okay? And here is mutation 3, translocation. Part of it broke off and attached to a different chromosome. I believe that is all of it. Let's just compare and contrast mitosis and meiosis. So mitosis, it results in two identical daughter cells that are diploid, okay? Whereas in meiosis, it results in 4 genetically different haploid cells. Okay, so these would be somatic cells. It makes somatic cells, which remember are our tissue cells, muscle cells, liver cells. Whereas in meiosis, we are making gametes, which are reproductive cells. Let me just make the font size a little larger. The function of mitosis is for growth and repair. And meiosis, its goal is to make gametes, to make reproductive cells, to make cells to pass on genetic information. Mitosis, my daughter cells are identical to each other. Meiosis, my gametes are genetically different. In mitosis, also my daughter cells are genetically identical to the parent cell. And here they are genetically different. The gametes are genetically different from the parent cell. Okay. Meiosis, the cell splits 2 times, whereas in mitosis, the parent cell splits once. There's no crossing over in mitosis, whereas crossing over does occur in meiosis. All right. I'm going to say some similarities. They both involve the splitting of cells. They both involve the aligning of chromosomes at the metaphase plate and the splitting of sister chromatids. In mitosis, the splitting of sister chromatids occurs in anaphase, whereas in meiosis, the splitting of sister chromatids occurs in meiosis 2 or anaphase 2. It is slightly different, though, the splitting of sister chromatids, because in mitosis, those sister chromatids are identical. In meiosis, those sister chromatids are not identical because of crossing over. And yeah, I think